So, very good evening, my dear friends. Today's eminent resource person, Dr. K.RANI Madam Garu, Assistant Professor, Department of Botany, Government Degree College, Autonomous, Siddipet. Madam is going to explain one of the important area in plant physiology, that is photosynthesis. Madam, please start your session. Please share your screen. Start your session, madam.
Screen share. Sir, give me host, sir.
Madam, please share your screen. I have given to you option.
Okay, sir. Sorry, it will show us just like host disabled participant screen sharing, sir.
1 minute, madam. 1 minute. 1 minute. Okay, now it's
okay, sir.
Madam, please share.
Okay, sir. Okay.
Right.
Thank you, sir. So this is Rani from GDC Siddipet.
Madam, please enlarge the slides. Okay.
Okay. So today we are going to discuss about the C4 pathway. So last classes we'll discuss that C3 cycle almost in the photosynthesis. It is the second step, Nana. Dark phase. So dark phase is divided into 3 phases. So in that C3 is completed yesterday. Today we are going to discuss about that C4 pathway. Dark phase reactions in that C4 pathway. So what is C4 pathway? Generally, the plants, they are adapted to dry tropical regions. They have the C4 pathway mentioned earlier. So though these plants have the C4 substances, just like oxaloacetic acid as the first carbon dioxide fixation. So all these three steps, they are the CO2 fixation, carbon dioxide fixation. So in that C3 is one of the way, and this is the another way of the CO2 fixation. In this, the indication that there exists another pathway to C3 pathway came from the work of the Kortschak and Burr. So he is famous scientist Kortschak and Burr in 1961. They reported that in some plants, the first stable compound in carbon dioxide fixations, so that is C4 compounds like oxaloacetic acid in the C4 pathway. So oxaloacetic acid is the 4-carbonous compound. So that's why this pathway is known as a C4 pathway. So earlier we'll discuss that C3, in that the first stable compound is the phosphoglyceric acid. PGA, so that is the C3 compound, 3-carbonous compound, that is the first stable compound in the C3. That's why that is C3 pathway. So in this C4, the 4-carbonous compound is the first stable compound, that is oxaloacetic acid or malic acid or aspartic acid. So these 3 almost C4 compounds. So likewise, this is known as a C4 pathway. Okay, so then almost in what way are they different from C3 plants? This is almost a question that you may reasonably ask. The C4 plants as the special, Nana, they have a special type of leaf anatomy. Okay, so they tolerate the higher temperatures. They will show a response to high light intensities. So they can lack a process that is called photorespiration, and they have a greater productivity of the biomass. So just almost let us understand this one by one in this pathway. So after that, later after almost Kortschak and Burr in 1961, later MD Hatch and C.R. Slack. MD Hatch and C.R. Slack. So the 2 Australian biochemists, they will work out the details of the this C4 pathway. So that's why this is discovered's name by the Hatch-Slack pathway. C4, as per same, the another name of the C4 is Hatch-Slack, and it is also known as a C4 dicarboxylic acid pathway. So it is almost an alternative pathway to the C3 plants or C3 pathways. Okay, so what the names of that? Almost to C4 pathway. So these all are the alternate C4 plants. Okay, so the C4 cycle, it has been reported in 19 genera. Almost in the plant kingdom, it is reported in 19 genera, 90 species. They all are belonging to 11 families. So as per Bentham-Hooker classification, we know that almost 19 genera and 90 species belonging to 11 families. So in this almost majority are the monocots. So Poaceae and Cyperaceae, these all are the monocots, and some of the dicotyledonous plants like Amaranthaceae, Portulaceae, and Boraginaceae. So these all are the dicotyledonous plants. 19 genera, 90 species almost. So they belonging to the C4 plants. In this generally, the study of almost vertical sections of the leaves, one of a C3 plant and another of the C4 plant. They both are the different from the leaves anatomy. So almost do you notice the difference? You know that do both have the same types of the mesophylls? They are similar cells around the vascular bundle sheath cells. These all are same in the mesophyll tissue of the leaves, either C3 and C4. But the particularly in the C4 plants, some large cells, they are around the vascular bundles of the C4 pathway plants. So they are known as bundle sheath cells. Almost just like we'll take that monocotyledonous leaf anatomy in the upper epidermis, lower epidermis, mesophyll tissue. So it will not differentiate the palisade and spongy, almost all the spongy cells. So in the vascular bundles, it will almost the large cells around the vascular bundles are presenting in the C4 plants. So they are called the bundle sheath cells. So almost in this pathway, the primary, the atmospheric carbon dioxide acceptor is phosphoenol pyruvate. So I will show the almost cycle. This is the in the cycle, C4 cycle, you can observe here mesophyll cell and bundle sheath cells. So this is the almost these plants are from the okay. So during the primary carboxylation reactions, these plants, the CO2 combines with phosphoenol pyruvic acid. So first step in this cycle, the carbon dioxide, atmospheric carbon dioxide combines with the PEP, phosphoenol pyruvic acid, to form the oxaloacetic acid. So which is then it is reduced to malic acid. It is almost aspartic acid or malic acid or oxaloacetic acid. This is the 4 carbonous compound, Anna. So that's why this cycle is known as a C4 pathway. Okay, so this is very common or very important point in the C4 pathway. This first atmospheric carbon dioxide, it is almost combines with the phosphoenol pyruvic acid. So it is presenting in the leaf anatomy of the C4 plants, and phosphoenol pyruvic acid is combined with carbon dioxide to form the oxaloacetic acid. So then it is reduced to malic acid. In this way, almost in the carboxylation of the phosphoenol pyruvic acid, it is participate the enzyme phosphoenol pyruvic carboxylase enzyme. So you can observe here atmospheric carbon dioxide entering into the mesophyll cell, and it is combined with phosphoenol pyruvic acid to modified as a C4 acid. So that is the oxaloacetic acid. So in other way, in almost in these three different substances, you know that C4, oxaloacetic acid, malic acid, and aspartic acid. So it is different from other plants, combines with the in the carboxylation. So in this process, almost which enzyme is in the presence of phosphoenol pyruvic carboxylase enzyme is induced in this reaction. So the 4-carbon acid, that is almost, it is the dicarboxylic acid is hydrolyzed to pyruvate. So pyruvate with the simultaneous release of the carbon dioxide. So the carbon dioxide is then fixed into ribulose diphosphate. So it is, which is the part of the first reaction of the Kelvin cycle. So in other way, just I will discuss you. This is almost in this pathway, the CO2 acceptor is the phosphoenol pyruvate. So up to here, clear, Nana? And after that, the carboxylation is catalyzed by phosphoenol pyruvic carboxylase enzyme. Okay. So the first stable compound, that is the oxaloacetic acid. It is the C4 compound. So it is dicarboxylic acid. So 2 COO, almost 2 carboxylic acids, 2 are presenting in this compound. So that's why this is also known as dicarboxylic pathway. So in this pathway, both C3 and C4 pathways, both will operating in this. So this is the almost the C4 plant leaf with the Kranz anatomy. So it is an entrance point of view. What is Kranz anatomy? Almost we know that the particularly the large cells, you can observe here, large cells around the vascular bundles of the C4 pathway. So these plants are called bundle sheath cells, and the leaves which have such anatomy, so that is called Kranz anatomy. Kranz means, so it is in the Japan word wreath, Nana, WREATH. So Kranz means wreath, and it is a reflection of the arrangement of the cells. So bundle sheath cells and mesophyll cells. So in the center point, it look like a flower vase, just to flower vase type structure. We will observe here the blue color structure. So that is look like a Kranz anatomy. In the Japan word, Kranz means wreath. Wreath means flower vase or flower like structures. Okay. So it is a wreath.
Excuse me, madam. Madam. I hope it is the Germany word. Maybe is you to correct it. That is the Kranz.
Okay, okay. Sorry, sir. Okay.
Okay, no problem. Please, madam, proceed. It is a Germany.
Okay.
German. The term.
Germany. Okay, okay. So
okay. Please proceed.
Sorry for the interruption. So this is the Germany word, Nana. Almost Kranz means wreath. So reflection of the arrangement of the cells. Okay. So you can observe that is the almost in the center part vascular bundles. Vascular bundles are the particularly large cells around the vascular bundles. So that is bundle sheath cells. These cells look like a such anatomy, that is the Kranz anatomy. So Kranz means in the Germany wreath. So it is reflection of the arrangement of the cells. Okay. So the bundle sheath cells are characterized by a large number of chloroplasts. So in the bundle sheath cells also have the chloroplast. So the thick walls, they are impervious to gaseous exchange and no intercellular spaces. So almost the chloroplasts are the dimorphism in this way. So in the mesophyll cells also contain the chloroplast, and bundle sheath cells also contain the chloroplast. So the bundle sheath cells are the large number of chloroplasts, thick walls, they are the impervious to gaseous exchange and no intercellular spaces. So in this almost you may like in the cuttase section of the leaves, the C4 plants like maize or sorghum or any monocotyledonous, you can observe the Kranz anatomy, and they can distribution of the mesophyll cells also we will observing in the TS of the leaf. So this is the Kranz anatomy, Nana. Just to the all C4 plants, they can exhibit a specialized anatomy. So Kranz Anatomy, we know that. So all these plants, they can possess a bundle sheath made up of a large sized cells around the vascular bundles. So the chloroplast of the mesophyll and bundle sheath cells are the morphologically different. So almost the mesophyll chloroplast are the normal with grana, and bundle sheath cell chloroplast, they can contain the starch-rich chloroplast. So they are the bundle sheath cells lacking of grana, but mesophyll chloroplast are the normal with grana. So this is common one or two differentiation we'll observing in the chloroplast. So that's why the chloroplast in C4 plants are the dimorphic. All chloroplast in the mesophyll cell also chloroplast, and bundle sheath cells have the chloroplast, but they are the dimorphic in nature. Okay, so and next in the cycle purpose, the primary function of Kranz Anatomy, it is to provide a almost to carbon dioxide. It can be concentrated around the rubisco. So yesterday in the C3 cycle, we'll discuss that rubisco. So there is avoiding of the photorespiration. Almost in these plants, the C4 plants have there is avoiding of the photorespiration. Okay. So the mesophyll and bundle sheath cells are the connected through the numerous cytoplasmic sleeves. They are called plasmodesmata. So this is common in all the plant tissues, almost interconnected with one cell to another cells with the thread-like structures, that is called plasmodesmata. So in the same manner, the mesophyll and bundle sheath cells, all the cells are connected through some cytoplasmic thread-like structures. They are called the plasmodesmata. Okay. So in the C4 cycle, there are 4 phases are there. So in the first step is formation of C4 acids with the help of the PEP carboxylase in the mesophyll tissue or mesophyll cells. Okay. So almost C4 cycle is completed in 4 steps or 4 phases. So in that first phase is the formation of C4 acids like oxaloacetic acid or malic acid or aspartic acid. So these 4 are the 4 carbonous substances. So the formation of C4 acids with the help of the phosphoenol pyruvic carboxylase in the mesophyll cells. Okay. So almost you know that the just we'll discuss later. And second step is transport of C4 acids into bundle sheath cells. Almost so whenever the mesophyll tissue form the C4 acid, the C4 acid is transported into bundle sheath cell. And third step is decarboxylation of C4 acids to release the carbon dioxide, so which is fixed as a carbohydrate by C3 cycle. So in this almost in the third step, the C3 cycle also the part of the third step. And fourth step is C3 acid. So that is pyruvate formed in the decarboxylation. It moves back to the mesophyll and helps in the regeneration of the phosphoenol pyruvic acid. So this is the major 4 steps. So it is very simple, very important. That is the complete the formation of C4 acid. So it is transferred into bundle sheath cell and decarboxylation phase. And it is the move back into our regeneration of the phosphoenol pyruvic acid. Okay. So this is complete diagrammatic representation of the C4 cycle, Nana. In the first step, almost we'll discuss that the atmospheric carbon dioxide entering into the mesophyll cell. So the primary CO2 acceptor, it is a 3-carbon molecule, that is the phosphoenol pyruvic acid. Almost it is interconnected to some bicarbonates, HCO3. So phosphoenol pyruvic acid is very key enzyme. Almost in the C3 plants, we'll discuss that RUBP, Rubisco enzyme is the very active and key enzyme in the C3 plants. So as per same, in the C4 plants, PEP, phosphoenol pyruvate. So it is 3 carbonous molecule. It accepts the CO2 in the form of HCO3 minus, and that is presenting in the mesophyll cells. Okay. So the enzyme responsible for this fixation, that is the phosphoenol pyruvate carboxylase. So in simple way, pepcase. In other way, so it is also known as pepcase, phosphoenol pyruvate case. Pyruvate carboxylase or pepcase enzyme. It is very important. So important to register that the mesophyll cells lack the Rubisco enzyme. Almost in the C4 plants, it is lack the Rubisco enzyme. So the C4 acid, the PEP is combined with carbon dioxide. The C4 acid, oxaloacetic acid is formed in the mesophyll cell. That is the almost in the fixation. C4 acid up to C4 acid, that is the first step now. Okay, so atmospheric carbon dioxide entering through the stoma and entering into the mesophyll tissue of the C4 plant leaf. So in the mesophyll cells, it contains the PEP, phosphoenol pyruvate. So that is 3 carbonous molecule and combined with carbon dioxide, that is formed as a 4 carbonous substance, that is the oxaloacetic acid. So it accepts the CO2 in the form of the HCO3, and that is presenting in the mesophyll cells. And the second step, almost then the forms of other 4 carbon compounds, almost as I told you that malic acid or aspartic acid also in other 4 carbon compounds. So in the mesophyll cells itself, they are transported into the bundle sheath cells. So in the lower one, that is the upper one is the mesophyll cell and lower one is the bundle sheath cell, Nana. So the 4 carbonous compound is transported into the bundle sheath cells. So in the bundle sheath cells, these C4 acids are broken down to release the carbon dioxide and a as per 3 carbon molecule. So మనకి పైన అయితే ఎట్లా అది కార్బన్ డైఆక్సైడ్ తోటి కంబైన్ అయిపోయిందో మళ్ళీ ఇక్కడికి వచ్చేసరికి అంటే బండిల్ షీత్ సెల్ కి వచ్చేసరికి ఆల్మోస్ట్ ఇన్ ద బండిల్ షీత్ సెల్స్ దీస్ C4 acids are broken down to release the carbon dioxide and other 3 carbonous substance or 3 carbonous molecule. So the 3 carbon molecule is transported back to the mesophyll where it is converted to phosphoenol pyruvate again. So thus completing the cycle. So in other way, in the almost in the bundle sheath cell, the released carbon dioxide. So this is almost you can observe here decarboxylation phase to C3 acid. So one CO2 is eliminated, or decarboxylation means one CO2 is eliminated from this step. So the released CO2 in the bundle sheath cells, they will entering into the C3 cycle or the Kelvin pathway. So this pathway is common to all plants. Not only it is restricted to C3, Nana. C3 is involving in the C4 and CIM also. So that's why the C3 pathway is common to all the plants. So the bundle sheath cells are rich in the enzyme, that is the Rubisco. So now mesophyll cell contain the only PEP enzyme, but bundle sheath cell, chloroplast or bundle sheath cell containing the Rubisco, ribulose, biphosphate, carboxylase and oxygenase. So that is completely bundle sheath cell have that enzyme. So that's the basic pathway that results in the formation of the sugars also. So the Kelvin pathway is common to C3 and C4 plants. That is the main concept of this cycle. So almost you know that, did you know that the Kelvin pathway occurs in all the mesophyll cells of the C3 plants? So it does not take place in the mesophyll cells, but does only in the bundle sheath cells. So that is the C4 importance or C4 cycle of the dark phase. So what is the, almost we know that the C4 plants have important 3 characteristics. So what are they? Almost the unique distribution and kind of chloroplast found in the mesophyll cell and bundle sheath cell. So the dimorphism of the chloroplast. Chloroplast is, it have the dimorphism. That means it is the unique distribution and some special kind of chloroplast found in the mesophyll and bundle sheath cell. This is the important characteristics of the C4 plant. And another important, the high concentration of certain enzyme like phosphoenol pyruvic carboxylase in the leaves, in the C4 leaves. So the C4 leaves have the, which enzyme nana? Phosphoenol pyruvic carboxylase is presenting in the high concentration of the high concentration of certain enzymes. They are presenting in the C4 plant leaves. And third important character, so the ability to fix the carbon dioxide at extremely low concentrations. Okay, so the ability, it will fix carbon dioxide at extremely low concentration of the carbon dioxide. So these 3 main characteristics of the C4 plants. So through the plant breeding experiments and some genetic manipulation also, it will be possible to produce the agriculturally useful plants, so that can grow in hot to arid environments also. That is the other important of the C4 plants. Okay, so almost this is the Kortschak pathway. And so the types of the C4 plants, Nana. C4 plants, 3 different types of C4 plants are recognized. So it depending upon the C4 acid. As I told you that malic acid, aspartic acid, and some oxaloacetic acid. So these all are some types of the C4 plants. So almost in the plant, the 3 different types of plants are recognized. So it is depending upon the 4 carbonic acid transported to bundle sheath cells, and the nature of the enzyme involved in the decarboxylation reactions in the bundle sheath cells. So in that first one, NADP-ME type. So Malate to farmers, example, sugarcane and maize. See, both will be monocotyledonous almost. And second one is the NAD-ME type. So that is aspartate farmers, for example, Amarantaceae family, Amaranthus. And PEP type, so that is carboxylase type, that is panicum and chloris. So these all are the types of the C4 plants. So almost, hello sir, how much time will remaining?
Madam, it's 6 minutes.
Okay, so the CAM cycle almost shall I start or tomorrow we'll discuss?
Madam, CAM pathway tomorrow we'll discuss, madam.
Okay, okay. So any doubts regarding C4 cycle, my dear students? So almost in the examination point of view, what is Kranz anatomy? So almost sir said the Germany word, sorry, I forgot, almost Germany word Kranz. So it is look like flower type structure, and chloroplast dimorphism. So what is the structure and shape of the bundle sheath cells? This is very important. And what is the first stable compound of the C4 cycle? So which enzyme is key enzyme in the mesophyll cells? That is the PEP case. Almost C3 also completed.
Madam,
what is that?
Madam, it is visible C3 and C4 pathway differences.
Okay. Okay.
Madam, please, if possible, please explain these differences between C3 and C4 pathway.
Okay, sir. Sure, sir. Sure. Thank you. So this is almost C3 cycle also. We'll discuss in last classes. C4 today. What is the difference between the C3 and C4 cycle? So almost the photosynthesis is occurring mesophyll cells. only mesophyll cells also, it will happen in the C3, but in the C4 plants, it occurs the mesophyll and bundle sheath cells. So the chloroplast dimorphism we'll discuss just now. So the photosynthesis occurs in the mesophyll and bundle sheath cells. And the second point, so the primary acceptor of the CO2 is the RUBP in the C3 plants. So the C3 plants in the mesophyll tissue, Rubisco enzyme is very active enzyme. So that is very key enzyme. Rubisco is presenting in the C4 plant leaf anatomy. So that's why the CO2 molecule acceptor is the RUBP. So, but in C4 plants, the CO2 acceptor molecule is PEP, phosphoenol pyruvate. Okay, so in the C3 Rubisco, in the C4 PEP, phosphoenol pyruvate. So in the first stable product in the C3 cycle, that is the 3-carbonous compound, Nana, that is called phosphoglyceric acid, PGA. So that is the 3-carbonous compound. That's why this cycle also known as C3 pathway. As per same in the C4 cycle also, the first stable product, that is the 4-carbonous compound, that is called oxaloacetic acid. Okay, so 3-carbonous compound C3 cycle, 4-carbonous compound C4 cycle, 4-carbonous oxaloacetic acid. So in C3 plants, the photorespiration rate is high and it leads to loss of fixed carbon dioxide. It decreases the CO2 fixation rate. So this is almost in the photorespiration occurs in C3 plants, but in the C4 plants, photorespiration is not found. So it is negligible and it is almost absent in the C4 plants. So hence it increases the CO2 fixation rate. So whenever the availability of CO2, that is almost in the C3 plants, we'll discuss that Rubisco. Rubisco also act as a carboxylase and oxygenase. So for that purpose, the photorespiration rate in high, it leads to loss of the fixed carbon dioxide. So it decreases the CO2 fixation rate. But almost in the C3 plants, the optimum temperature is 20 to 25 degrees centigrade. So that is almost in the room temperature. But C4 plants are the arid, almost to the optimum temperature. Just comparable to C3, it is just high, 30 to 45 degrees centigrade. Almost monocotyledonous and chepukuntam. So the optimum temperature of the C4 pathway is 30 to 45 degree centigrade. So in the C3 plants, that is example of C3 plants are the rice, wheat and potato. Almost in the C4 plants, that is IS2 monocam, maize, sugarcane, tribulus, amaranthus and other plants. These all are the C4 plants. So in the C4 plants, almost they are the some dicotyledonous plants also have. So Amaranthaceae and Portulaceae, these all are the C4 pathway plants. That is maize, sugarcane. You know that in the leaf anatomy part, so almost monocotyledonous leaf and dicotyledonous. So that is differencing in the mesophyll tissues of the C3 and C4. These all are some other difference between the C3 and C4 plants. In this, almost the CO2 fixation rate under highlight conditions in the characteristics of the C3, C4, some take